Welcome to today's lab. We'll be investigating the enzyme catalyst, which can be found in potatoes, and we're gonna be placing the potatoes in different environmental conditions. This is in room temperature. At room temperature, frozen potatoes, heated potato, boiled. This has been treated with vinegar, which is an acid, and the last one has been crushed with the pesto. Um, we will be using hydrogen peroxide, which you can actually like put on wound, and it has that bubbling sensation because the hydrogen peroxide uh, is broken down by an enzyme in your body called catalase. So catalase is also found in potatoes. So we have extracted a piece of the potatoes by using the straws, getting them out, and we cut them in different pieces and place them in different containers. Three potatoes were already added to this test tube. These are the room temperature potatoes. Three were also added to the cold one. And what I'm going to do next now is to add 5 milliliters of the hydrogen peroxide, which I store in brown bottles so that it doesn't decompose into water. So I'm just going to use the pipette and quickly add this. And after adding it, we're going to set the timer to 5 minutes. Oh, I see some reactions happening. You guys see the foam? Can you look closely? Yeah, some foams actually happen. All right. So room temperature is done. I'm gonna add some hydrogen peroxide to the frozen potato. And I set the timer beginning now. So this is going to run for 5 minutes, and after 5 minutes we will use the metric ruler to measure the height in centimeters, and I will record the data here. We'll pause for a bit and come back after 5 minutes. After 5 minutes we measure the height of the foam. The one for the room temperature potato is 1.1 centimeter, and the one for the frozen potato is 1.2 centimeters. We know that this is the winter time and the temperature um, outside and inside is also very cold. So that's why we have the almost similar data. Now the next part of the experiment will be putting the boy and the acid treated potato in these test tubes. So we're gonna pause, and I'm gonna like clean them and add these two into the tubes, and then we'll do the same experiment for 5 minutes. Welcome back. Pouring some hydrogen peroxide in the container. You want to keep it close because if you kind of like put it early, it's going to decompose into water, so it wouldn't give you a good result. So now we've already added the heated potato to the test tube on the left, and the one treated with vinegar has been added to the one on the right. So we're going to add equal amount of hydrogen peroxide using a dropper, 5 milliliters. I want to see after 5 minutes what is going to make foam. I'll measure using the metric ruler. All right. I'm going to set our timers for 5 minutes. And I'm going to take a pause and come back after 5 minutes. Welcome back. So as you see, after 5 minutes, there's no foam, but by the potatoes that has been treated with heat, and also the one treated with the acid, the vinegar. So that tells us something happened to the enzymes in the potatoes. So our measurement will be 0 centimeter and 0 centimeter. Then the last part of the experiment, I want to do these two together, the room temperature and the crushed potato, because this is the last one we haven't done yet. So I need to watch these two tubes. Even though we did the room temperature, I just want to compare it with the crushed one. So we're going to take a break now and come back. Okay, welcome back to the last part of the experiment. So we are going to take the potatoes and crush them. So by crushing the potatoes, it increases the surface area of the enzyme to react with the substrate, which is hydrogen peroxide. I've already added the crushed potato to this test tube. It has been added to this. And just for reference, I am adding, this is our control. The room temperature potato has already been added. As you can see them at the bottom of the tubes. So I'm going to add the equal amount of hydrogen peroxide to the first one, which is our room temperature potato. Initially we had the height of the foam was measured to be 1.1 centimeter. So I've added 2 millimeters, 3, 5 millimeters, and we see some reactions happening. Then the one with the crushed potato, I'm adding the same amount. I'm going to set our timers for 5 minutes. Wow, I'm noticing a lot of reactions happening with the ones that has been crushed. The foam is almost double the one with the room temperature potato, and it hasn't been 30 seconds yet. So this is going to go on for 5 minutes, and after 5 minutes we're going to write our data for the crushed potato on this side. So we're going to pause the video and come back after 5 minutes. Welcome back. The height of the crushed potato is, wow, about 2.2 centimeters. And this one is still 1.1. So we write our data, 2.2 centimeters, and this is still 1.1. So our substrate is hydrogen peroxide. The formula is H2O2, and our potato contains the enzyme catalase. So catalase the enzyme can be found in all living things, including potatoes. So when you expose potatoes to different environmental conditions, it affects the production of the foam. So the enzyme breaks down the substrate into water and oxygen. I hope you guys enjoyed this experiment, and I'll see you another time.